Welcome back, everyone, to our continued discussion of functional neuroanatomy. Um, Today, we're going to talk a little bit about development, about how the brain develops, how portions of the neural tube become, the parts of the brain that we talked about and know, and just what these different parts do. We'll start to get some of that differentiation. So with that, we're going to start with this video. Again, I'll provide you a link in the description of prenatal brain development, which will give you a really nice overview about how this starts, and then we'll kind of carry it through to how, what that development ends up working into.
This animation shows brain development from conception through birth. In the first 4 weeks after conception, neural tissue known as the neural plate forms in the outermost layer of embryonic cells. The neural plate folds to form the neural groove, then curls to form the neural tube. This tube differentiates into the forebrain, the midbrain, the hindbrain, and the spinal cord. The forebrain will develop into the cerebral cortex, which translates sensory stimulation. It also controls complex behaviors, thoughts, memories, and problem solving. The midbrain will develop into a neural relay station for sending information from the body to various sites in the brain. The hindbrain will control the most basic physiological processes, such as breathing and heart rate. The spinal cord is the pathway for conveying information between the brain and the rest of the body. Between weeks 4 and 8, the embryo grows rapidly and the face becomes recognizably human. The eyes on the side of the head begin to migrate forward and two distinct hemispheres of the cerebral cortex emerge. Between weeks 8 and 26 of the fetal stage, the cerebral cortex grows to cover the midbrain. By week 28, a major structural change begins. The cortex expands greatly in surface area and becomes wrinkled and folded inside of the skull. Beginning at week 28 and continuing through week 40, the brain surface fills with hills and valleys, called gyri and sulci. These cerebral folds and wrinkles help to create a vast neural network, which allows the brain to become a highly flexible yet precise control center and processor of information. By week 8, the structures of the brain are already in place, but the amount of brain tissue is rapidly increasing as many new nerve cells are formed. The formation of new cells is known as neurogenesis or neural migration. During neurogenesis, new nerve cells migrate from the innermost layers of tissue outward. Like gymnasts climbing ropes, the young cells are guided to their final destination by fibers from another type of cell, the glial cell. Glial cells form the underlying structure of the nervous system. Although earlier formed cells rest closer to where they originated, later formed cells move further away from their starting point. In this way, the brain grows, adding layers to itself like the skins of an onion. Once each nerve cell reaches its destination, it begins to acquire characteristics specific to that area of the brain. At this point, each nerve cell sprouts the extensions it needs to communicate with other neurons. There are two types of extensions, dendrites and axons. Dendrites look like a network of antennas and receive incoming signals. An axon is a single, larger extension which carries outgoing signals to other neurons. Synaptogenesis is the process in which synapses proliferate in the brain. A synapse is a tiny gap between the dendrites of one neuron and the axon terminal of the next. It's the point of communication between two neurons. So many new axons and dendrites form during the process that it's sometimes called exuberant synaptogenesis. This cell growth process continues into a newborn's first year of life. For an information signal, or impulse, to move from the axon of one neuron to the dendrites of another, it must cross the synaptic gap. In this animation, an impulse looks like a green ball of light. When an impulse reaches the end of an axon, a chemical called a neurotransmitter is released. In this animation, neurotransmitters look like red balls of light. Neurotransmitters allow an impulse to cross from the axon of one neuron across the synapse and onto a receiving dendrite on another neuron. To create a more efficient and rapidly working brain, some of the newly formed synapses are eventually removed. Synaptic pruning occurs during the fetal period, infancy, childhood, and adolescence. It's believed that pruning is a way in which the brain becomes customized to an individual's nervous system as a response to that individual's unique experiences.
All right. So a nice summary of development and also a nice little review of last lecture. So let me get the slides up. Here we go. So as was discussed in the video, in the first month after conception, the neural tube emerges, and in the neural tube, you have the areas that are eventually to be called the forebrain, midbrain, and hindbrain. And these sections are subsequently further divided as they develop. So you can see here, within 25 days after conception, so very early, you have this neural tube with these, forebrain, midbrain, and hindbrain. And they very quickly grow, as you can see. And then further they'll specialize and further develop, as you can see here, where the forebrain has become the whole outer cortex. Midbrain has really tucked itself under the forebrain, and then hindbrain is starting to differentiate into things like the cerebellum, the pons, medulla, things of that nature. There are also a couple developmental markers in the middle. And let me actually go to the next slide. I think it'll be a little easier to grasp how development works on that slide. So this is actually a figure from the book, and I highly recommend knowing it. Study this, know this, you will thank me later. So this is really going to tell you how things develop and what they break into. So with this, as you can see, what starts as the forebrain ends up developing into the regions both at the front and the back of the brain. So don't let the name fool you, because it's a large area. It becomes, much of the, not only the cerebral cortex, but many of the other systems too that are throughout the brain. So the forebrain gets divided into two areas after about 50 days of development. You have the telencephalon and the diencephalon. So here you see telencephalon, diencephalon. Telencephalon will eventually develop into the cortex, basal ganglia, and the limbic system, whereas diencephalon will be the thalamus and the hypothalamus. The midbrain, its next stage of development is the mesencephalon. At this point, its development stops. And then you have the hindbrain, which breaks into the metencephalon, and then you also have the myelencephalon. This myelencephalon will become the medulla. Metencephalon will become the cerebellum and the pons back here. So that's the hindbrain. And then, of course, you also have the spinal cord, as was mentioned, coming out of this as well. So you see that, kind of stage development, where first you just have forebrain, midbrain, hindbrain. Then you have these kind of middle stages of development, and then it begins further specializing. So if I were to study this, what I would really look at is what parts go with what stage of development and what overall route. So cerebellum was, originally part of the hindbrain, things like that. So now, like we did with the neuron, we move from focusing on its form to discussing the functions of the brain. So before we get into specifics, there are a few things worth noting about the brain. First, with the exception of the corpus callosum, pineal gland, and pituitary gland, most structures of the brain have both a right and left hemisphere. So there's 2 copies of almost everything. Secondly, as is the case with all vertebrates, each hemisphere of the brain controls the opposite or contralateral side of the body. Thus, the areas of the brain responsible for movement on the left side of the body allow me to use my right arm. So left side controls the right side, left side of the brain, that is, controls the right side, and vice versa. I just realized that these motions mean nothing because it's a mirror image, but left side of the brain, right side of the body, and vice versa. The basal ganglia, which we'll be talking about a fair amount throughout the class, but especially in this chapter, is made up of several structures which we will discuss momentarily, and it's important for motor control, procedural learning such as tying one's shoe, and certain habitual behaviors. Since it's important for behavior, problems in the basal ganglia are associated with several movement disorders such as Parkinson's disease and Huntington's disease. It's also been shown to be related to some disorders of behavioral control, such as Tourette's syndrome and obsessive-compulsive disorder. So as you can see, the basal ganglia sits beneath the cerebral cortex, and it's made up of 4 structures: the caudate nucleus, the putamen, the globus pallidus, and which is in the telencephalon, and then the substantia nigra, which is in the midbrain. These structures are connected to each other as well as to the cerebral cortex, which creates sort of a looping neural system. We'll learn a lot more about the basal ganglia in Chapter 11, but for now you just need to know it's important for movement and know those 4 structures that make up the basal ganglia. As a clinical psychologist, the limbic system is one of the most important systems that we deal with, as it plays an important role in emotion regulation and in emotion in general. It's also very important for learning and memory. So as you can see, the limbic system is also made up of several different structures, but unlike the basal ganglia, these structures are much more spread out and not as strongly connected. So looking at some of these structures, the amygdala is one that's very important to psychology. As you can see, it's responsible for an assortment of things, including emotion regulation and perceiving odor. Given the emotional regulation component, this is a structure that's extremely important for clinical psychology. The hippocampus and phonics are associated with forming new memories and learning. The easiest way to remember this is with the hippocampus. If you saw a hippo on campus, you'd probably remember it. So hippocampus, very important for learning and creation of new memories. The cingulate gyrus, like the amygdala, is responsible for many different activities. But the primary one, as far as you need to know, with its response and responsibility to the limbic system, is it's responsible for focusing attention, which is very important for learning and memory. And lastly, the olfactory bulb, as one might guess, is responsible for the sense of smell. And its relation to the limbic system kind of makes sense. When you think of a lot of the things, either things we learn or things we associate, a lot of times smell is very strongly associated. There are many things that you have a certain smell, and it reminds you of grandma's house for whatever reason, and you think of all the things that are associated with that smell. Really interesting how that works, but it makes sense given that it's part, it's linked in with the rest of the system. So limbic system is extremely important in terms of psychology. There are also a couple portions of the limbic system that can be found in the diencephalon. So the thalamus, which is near the center of the adult brain, acts as a way station to the cerebral cortex. So almost all sensory information enters the thalamus, where they're then sent to the appropriate sensory areas. Thus, the thalamus is an extremely important brain region to know about, as it's extremely important for perception. The hypothalamus lies directly beneath the thalamus, and it's responsible for many of our drives. It's been implicated in thirst, hunger, temperature regulation, reproductive behaviors, and more. The hypothalamus also controls the pituitary gland, which controls almost all hormone secretions. So again, very important for clinical psychology. So now we're going to look a little bit more at the midbrain sensory systems. So the most prominent feature of the midbrain are two pairs of bumps on the dorsal surface of the midbrain, one on each hemisphere. The more rostral, which again is toward the front, I put these on here just to help reinforce last lecture. Rostral toward the front, caudal toward the feet, so it kind of does this bendy thing, which is confusing. So rostral, the one that's more rostral is the superior colliculus, and this is important for processing visual information. The more caudal, so again toward the feet, boom, is the inferior colliculus, which is important for processing auditory information. So superior is visual, inferior is auditory. Together, they're known as the tectum. A couple other midbrain motor systems, some that are important to know. Substantia nigra, we mentioned briefly earlier, stands in Latin for black substance, that's how it's translated. And it's called this because its appearance is darker than most of the neighboring structures. So the substantia nigra is part of the basal ganglia. We'll be talking about this a whole lot more because it's really important for movement, as the neurons in this substantia nigra release dopamine. And dopamine is a very important neurotransmitter for movement. And it's thought that the death of these dopaminergic neurons are responsible for Parkinson's disease. So we'll come back to substantia nigra a lot. The red nucleus, which as you may guess is red in appearance, is important for movement because it communicates with the motor neurons in the spinal cord. Also in the midbrain is the reticular formation. It's a rather large structure that stretches from the midbrain all the way to the medulla in the hindbrain. Although the book is correct that the reticular formation is important for several functions, including motor and temperature control, it's probably best known for its association with sleep. Actually, given this, damage to the reticular formation can result in a loss of consciousness that does not end. So it's an eternal coma resulting in death. So it's a very important brain region. So now we're going to get more into the hindbrain. So at the base of the brain, we see the cerebellum, pons, and medulla. The cerebellum is primarily known for being involved in motor control, though it may also be important for some cognitive functions, such as attention and language, though that's a little less clear. It also helps integrate some sensory and motor functions together. The pons are located next to the cerebellum, and they're important for motor control and sensory information, in part because they include several cranial nerve connections. And then the medulla oblongata, also just known as the medulla, is at the base of the brain at the top of the spinal cord, and it contains several cranial nerve connections, including that control the neck and the tongue. It also contains a nuclei for controlling breathing and heart rate, so damage to the medulla is often fatal. So going back to the cortex, again, the part of the brain that surrounds, we're talking about the lobes again here. Um, so there are actually 6 distinct layers that are distinguished by the type of neuron and the pattern of dendrites and axons. For instance, the outer layers are much less densely packed with neurons than the inner layers. There are also some differences in the layers in different parts of the brain. However, for instance, I should say, layer 4 of the cerebral cortex is especially prominent in sensory areas. So layer 4, especially in the sensory areas, whereas layer 3 is especially notable because it has many large neurons. So they differ some depending on where they are in the brain, and also depending, some have certain neurons that are more prevalent than others. So these are some of the ways to look at the different layers of the cortex. So one of the cells that's really important are these pyramidal cells. So they look kind of like this. You can see how they get their name. So blown up, they look more like this, but you can still see the shape. And these are a class of large neuron that are prominent in the cerebral cortex. They're especially prominent in layers 3 and 5 of the cortex. And with the pyramidal cell, one dendrite of each pyramidal cell, called the apical dendrite, extends to the outermost surface of the cortex. So it goes way to the outside of the cortex. There are also several dendrites called the basal dendrites that spread out horizontally from the cell body. So here we have the apical dendrite. So this goes all the way up to the edge of the cortex. And basal dendrites, these spread out. So you have your pyramid shape. Make sense?